So as we start part 2 of chapter 3, we're going to get into more of the processes of the cells. So we looked at the cell structures with part 1. We're going to talk about how cells actually move stuff into and out of the cell. So that we usually generally refer to as just transport through the membranes or transport in and out of the cell. And you remember from part 1 that the cell itself has got this membrane around the outside. And the membrane is the gatekeeper of the cell. It's going to control what goes in and out. So you've got to get stuff past that membrane. And in some cases, it's a matter of like opening or shutting a door. You've got this channel that things can pass through. In other cases, they can just pass straight through the wall. So you've got the cell membrane made of those phospholipids. Things can pass all the way through it. And then in some cases, the cell's going to have to actually put forth a little bit of effort and expend energy because it wants whatever that substance is to come in, or it needs it to leave. So it's going to have to use a little bit of work. So we basically divide this down into like two big categories with subcategories underneath it. The first type is called passive and the second is active. And they're really, as their name sort of implies in here, passive means that the cell isn't having to expend any energy of its own for that. Active means the cell's going to have to put forth a little bit of work, so therefore it's going to have to expend a little bit of energy. It's actively involved in the transport of the substances across the membrane. So as I stated, passive transport then is going to require no energy, and that really should probably say no cellular energy. It's not that there is no energy involved. The cell itself isn't expending energy. All molecules have their own kinetic energy because they're made up of atoms, and atoms have those electrons that flow around the outside. So that's an energy in its own right, but it's not the cell's energy, it's the atom's energy. And so since those particles, those little atoms, have that ability to have energy from the electrons, sometimes that can be something that's useful, and that can be used for movement of atoms. For instance, if you spray perfume right here in front of your face, you're the first one to smell it, but eventually somebody across the room is going to pick up the whiff of that perfume because nobody's moving it. You're not blowing that over there. Even if the fan's not on in the room or whatever, it's not moving that there. What's happening is those little particles, those little atoms of the perfume are moving around. And as they move around, they start to bang into each other, and they're moving because those electrons are spinning around the nucleus of the atom. And then we bang and they start to push apart, and they push apart further and further and further. So the atom themselves has energy. So in passive transport, it kind of utilizes that own kinetic energy of the atom without the cell expending any of its own energy. So it doesn't have to make ATP for this, okay? We're also going to refer to concentration gradients through this whole thing. Concentration gradient just means where I have a lot, like where I first sprayed that perfume is a lot. That's a high concentration. The rest of the room would be low concentration because there's none. So it always goes from where there's a lot of it to where there's little until it's kind of evenly dispersed around the room. Molecules never stop moving. They're still going to move, but then there's no like net movement from high to low. So in passive transport, then you're always moving from where it's high to where it's low. It's kind of like flowing downhill. Here's the high amount. Here's the low amount. Molecules, if you had a ball or something at the top of the hill right here, it would flow down there and you wouldn't really have to put any effort. You just put it there and it's going to automatically flow. Eventually what's going to happen to that concentration gradient is it's going to equal out so that you have equal amounts of substances all around the area there. And there's these different types: simple diffusion, facilitated diffusion, osmosis, which is also a type of diffusion, and filtration are all types of passive transport that require no cellular energy. Active transport, on the other hand, does require the cell to expend some of its energy, and it's going to require that in the form of ATP. So those little mitochondria that we talked about, the powerhouses of the cells, they're going to have to make ATP, and the cell's going to have to expend that ATP in order to do this work. And the reason they're doing that is it's going in the opposite direction. So where we had in passive this high to low, you're actually moving uphill. So I have a low concentration of whatever it is, and I want even more of it. So I'm going to move that towards where I already have a bunch, and I'm going to have to force it. Okay, so the cell has to put forth effort to force it to go against that concentration gradient. We have active transport is a large category. You have also active transport that's kind of like a subcategory, so it kind of gets a little bit confusing, but we're going to talk about different types of this active transport. There's endocytosis, exocytosis. Remember, cyte means cell, and then transcytosis also involved in this. All of these are types of energy requiring or cellular energy requiring processes. Let's start with passive. First one that I mentioned was simple diffusion, and simple diffusion really is that. It's that example of the the perfume that I talked about, or somebody's barbecue in the neighborhood that you can smell from streets away. This is simple diffusion. It's going from where it started, it originated, there's a lot of it, to where there's none, the rest of the neighborhood in there. There's also a specific type of simple diffusion that we refer to as dialysis. Dialysis just really requires some sort of membrane to cross, where a barbecue is just passing those smells, those molecules through the air, little chemicals through the air. There's no barrier. Dialysis is simple diffusion with the addition of a permeable membrane. So now I've got a little bit of a barrier to get across, like a cell membrane, okay? So when we're looking here in these little beakers that I show you here, they're progressive. The green membrane, permeable membrane, just means things can pass through it, okay? Each of these molecules, you have little blue spheres and you have little red spheres. Each of them has their own concentration gradient. Since I have up a whole bunch of red on the A side, they're going to want to move to where there's none or less, which is the B side. And they do, as you look through the pictures here. The A's moving through, and it's moving through until eventually it's sort of equal concentrations. Doesn't mean the molecules stop moving. They'll still cross, but they cross in equal amounts. Likewise, the little blue spheres will also move at their own concentration gradient, and the blue ones are primarily on the B side to start off with, so they're going to move the opposite direction until they spread out kind of equal in there. So this simple diffusion really requires molecules that are pretty small, like O2 is just two atoms right there. Carbon dioxide is 2 atoms, and things that can easily pass through that phospholipid membrane. So lipid-soluble substances that don't have a problem getting across those phospholipids. Remember, it's passive, means there's going to be no energy from the cell that's being used here. Facilitated diffusion is still diffusion, so we're still not talking about no energy from the cell. But in this case, you have this extra term that says facilitated. Facilitated means the stuff can't pass right through these phospholipids. That would be simple diffusion. So this is simple right here, going straight through the phospholipids. They're too big, or they have a charge, or there's something that's preventing them from going through that membrane. So they need help. That's what the facilitated refers to. They need help. And their help comes from a protein, some sort of like channel protein. So things can flow through, and they usually flow through in one direction or the other direction, but they can flow through those channels. So they're getting help from a protein, but they're still flowing through a channel, and they're still not using energy in here. So it's not like the protein channel is having to do work. It's still going with that concentration gradient. It's still going from high to low, right, down that gradient. Larger molecules, like I said, some charge sometimes. So glucose is a larger molecule. Remember, it's C6H12O6. So it's a fairly large molecule. It's going to need a little bit of help. It can't just sort of flow through there. Charged things that have a charge. Amino acids are the basic structure of a protein. They're also pretty large, so they're going to have to use this facilitated diffusion. Osmosis is also a type of diffusion, but it is a very specific type of diffusion. So again, since it's diffusion, we're not talking about any cellular energy here. This is a diffusion that only refers to the movement of water. And water is, H2O. It has that polarity, so it has a little bit of a charge. That's going to prevent it from really moving freely through membranes. It does need a little bit of help from what we call aquaporins. Aqua refers to the water. Porins are little proteins, like in facilitated diffusion. Aquaporins help the water get through the membrane. But once again, we're still going with that concentration gradient from where it's high to where it's low. So there's no cellular energy here. Anytime you see the word diffusion or something that refers to diffusion, like osmosis is a type of diffusion, think no cellular energy. It's passive. The solutes can't, in this case though, we've got a special setup. So here's my beaker, here's my membrane, but where previously it said permeable, this one is selectively permeable, meaning that some molecules aren't able to pass through. So in this solution, we have a solution right here, and a solution is just a solute plus a solvent. Now water is the solvent, okay? It's what things are mixed in. Kind of think that like Kool-Aid. And then the solute is what I'm mixing in. So that would be the Kool-Aid mix, or the sugar, or whatever is the solute. The solvent is the water that you're mixing it into. So any solution is just made up of those two things, solute and salt plus a solvent. In this case, we have water. Water can freely move through this membrane, but our solutes, for some reason, are blocked. Okay, they can't move through. They're too big, they're charged, something's happening here. So the only thing that can move to try and get this at equilibrium, remember we're moving concentration gradient until you get to equilibrium, and it's at equal. Equal means equilibrium, okay? So we're trying to get where there's equal amounts on both sides. Well, unfortunately, in the case of osmosis, those solutes can't move. They can't go to equilibrium. So these big purple little spheres right here can't move across to equilibrium. They're stuck on one side. The only thing that can try and rectify this situation and make them equal concentrations on both sides, or equal dilution of the solution, so we're looking at trying to dilute to equal amounts, okay, is the water itself. So the water is going to have to do the moving. What happens is the water rushes to the side where there's more solute. So if you look here on side A, I have a whole bunch of these little purple spheres, and B, I don't have as much. That means B is less concentrated or more dilute. A is more concentrated, okay, which means it's less dilute, okay? So we're going to try and dilute it out. So the water moves across to dilute, but since the other particles can't move, look what happens to the amount of water. As water rushes across, you increase the water on side A and decrease on side B. But eventually, these little purple molecules are all equally spread apart from each other on both sides of that membrane, okay? So you're losing water from one side and gaining it to the other. Okay, this movement is going to be determined by something we call osmotic pressure, okay? So that's the ability of water to move through that membrane to try and dilute one side to make it equal to the other side, all right? And that's going to be a pressure amount. How much can I move over here before, gravity and everything else takes its toll? So as the water moves through, this has a bunch of solute particles in it, and then pure water would have zero solute particles. It's going to move through. It's going to change the amount of water on each side. The difference in those two is what we call osmotic pressure, okay? Osmotic pressure increases as the concentration of things that can't move through, non-permeable substances, increase. So you have a really high osmotic pressure on this side, right? And you have a lower on this side, and it's increasing here. So it's kind of getting to where it's trying to go to equilibrium, and it's going to move in an attempt to try and equalize both sides. It may never get there, but it's attempting to do so. All right, so what we need to do then is we need to define some terms. We look at the concentration of water, and we could look at the concentration of the solute, okay? So since a solution, remember, solution equals the solute, which it would be sugar, salt, ion, something, anything that's mixed in the water, right? Plus solvent, which is our water in most cases, in all cases when it comes to osmosis, all right? We need to look at how much solute there is compared to how much water. So it's a comparison thing. So this is what we're going to have to look at, and this is what we're going to have to name, all right? So in an isotonic, keep referring to this too, no cellular energy, right? All right, in an isotonic, iso means the same. So in an isotonic solution, the amount of solutes, these pink little spheres in this case, is equal on the outside as it is to the inside. Remember, molecules still move, so you'll have a little bit of water move in, you'll have a little bit of water move out, but it's equal amounts moving in and out. So the pressure is the same on both sides of the membrane, so there's no net movement. Even though you have water moving back and forth, there's no net gain or loss on either side of the membrane. It's moving in equal amounts, and the cell stays the same size. So the cell is not going to change size in this because there's no loss of water, there's no gain of water to the cell. In a hypertonic solution, and by the way, these refer to the amount of solute. So isotonic is the amount of solute that we have, okay? Same amount of solute on both sides. Hypertonic is a different amount of solute. Hyper means above. So this means I have a lot of solutes on one side of the cell compared to the other side of the cell. So my osmotic pressure is going to increase. So looking in the diagram right here, I have a whole bunch of these little pink spheres outside the cell. So this portion outside the cell is hypertonic compared, it's a comparison, compared to the inside of the cell. There's less solute on the inside of the cell. And what happens is we name the solution for the amount of solute. So the solution outside that cell is hypertonic. But remember, those little pink spheres, the little solutes, can't move. The only thing that can kind of move across the membrane to try and get this to equilibrium, right? We want to move to equilibrium as much as possible. The only thing that can move is going to be the water. So the water rushes out of the cell to try and dilute all of that solute out there, okay? The more solute there is outside the cell, the more water is going to leave the cell to try and dilute. So the greater the water loss, basically, from the cell. So what happens then is, since the cell is losing water to try and dilute the outside, it shrinks. And in animal cells, we call that crenation. So the cell gets smaller because it's losing that water to the outside environment. Here's another term, hypotonic. Hypotonic means it's low in solutes, okay? So if you look here, I've got a lot of pink spheres inside the cell, very few outside the cell. So the outside is hypo, it's low in solute. The inside is actually hyper compared to the outside, by the way, because it's a comparison, which means we have a lower osmotic pressure because we're low in solutes. We have the concentration of those solutes are more inside the cell. Again, the solutes can't move. The only thing that can move to try and rectify this situation is water. I have more water outside. This is high water out here, right? High water, low solute. This is high solute. Notice they're opposites. High solute and low water. So water moves to where there's a lot of water to where there's less water. We're going to move into the cell, and then that means that the cell is going to get bigger. It expands. And if it continues to get bigger and bigger and bigger, that cell membrane can only take so much pressure. It may actually burst, and we call this, it says hemolyze there, that's a verb. The noun is hemolysis. So it may go through hemolysis, or just simply put, lysis. Lysis means to break, okay? So looking at here, these are red blood cells. This is what a red blood cell in A should look like. So that would be equal amounts of water going in and going out of the cell. So the blood floating around in your plasma would be isotonic because there's some water going in, some water going out, but you're not like breaking the cell in any way. In the second one, you've got a lot of water moving out of the cell, so it's shrinking. That means that the outside must be hypertonic since water's moving out. So we have a hypertonic situation outside. Relatively speaking, then you've got a hypotonic initially situation on the inside, and water is moving out to where it's hypertonic, okay? And then in the third one, water is rushing in, the cell swells up. So that would be hypotonic out here, and the water moves in. In a hypertonic, remember that cell shrinking is called crenation. And then in a hypotonic, that cell swelling may cause it eventually. Right now, it's just swelling. You can just see it swelling. But if it continues and then it gets little holes in it and it bursts, that may lead to lysis of the cell. So it may lysis at some point. All right, there's another type of passive transport. This is called filtration. And filtration is kind of like your coffee filter. You filter the coffee through. You've got grounds, you've got water. The water washes through the grounds. The water is small enough to pass through that filter paper of the coffee filter, but the coffee grounds are not. They get stuck. So in filtration, that's exactly what you've got. Small molecules can move through. Large molecules are stuck on one side or the other. And this happens in our body also. This is a blood vessel down here. So as blood is moving through here, you've got all kinds of things in there. You've got red blood cells, you've got white blood cells, you've got proteins, you've got sugars, you've got water, you've got all kinds of stuff. The smaller molecules can actually pass through that capillary wall due to the pressure of the blood running through there, hydrostatic pressure. But the large particles never leave. They stay there, except special cases with white blood cells, and we'll learn that next semester. But unless, they have some action on them. But due to this hydrostatic pressure, you can only have the smallest molecules move through, and that would be water. So water leaves out through into the tissues, and it's going to have to be returned later back to the bloodstream. Another thing that works this way are the kidneys. So the kidneys, blood comes in, water and urea is forced out, and you form urine. And then the stuff that you still need in your blood and in your cells stays in the blood vessels and gets recycled back into the bloodstream. All right, active transport, remember, is requiring cellular energy. So we're going to have cellular energy being used here. That means that the cell has to make energy, it has to use energy. So the reason that it's doing this is because we're going against that concentration gradient. So a hill basically is a gradient. So we're going against that. We're going from where it's low to where it's already got a lot of molecules, and we want even more of those molecules to be shoved into that space. Okay, so from a low concentration to high concentration. Active transport, cellular energy. The cellular energy in many cases is going to be in the form of ATP. We're going against that gradient, like I said, does require that energy, and you need to usually think ATP. There's other energy forms that we can use, and we will use that with like ion gradients and stuff, but start kind of getting used to the term ATP. All right. Just like in facilitated diffusion, you are going to have to use proteins, but in this case, these are not like channels. They're not just like tubes or straws through the cell where things can easily pass through. These guys are called carrier proteins, and carrier proteins pump or force something through. So here's a molecule entering. It's going to use energy to push that all the way through, kind of like a water pump. You got to put pressure on that water pump. If you've ever been out on an old farm or seen that, they have to push to get the water through. It doesn't just flow through at ease. All right. So things that the cell needs, it might be sugars, amino acids, ions, because they're charged, so they can't pass right through here. They're blocked because of that charge in the center. It doesn't like it in the center of that phospholipid membrane. So ions are another thing that would use a carrier molecule. So again, unlike facilitated diffusion is more like a tube that it passes through easily, no cellular energy. Active transport is a carrier molecule, a carrier protein that does require cellular energy. So there's the difference between those two. One of the more common ones, it's like the poster child for active transport, is this thing we call the sodium-potassium pump. Sodium is an ion, potassium is an ion. What you have is we need to force a situation in nerve cells where there's a whole bunch of sodium on one side of the cell and there's a whole bunch of potassium on the other side of the cell. So I want to put as much sodium as I can out here and reduce the amount that's inside the cell. And the same with the potassium. I want to put all this potassium on the inside of the cell and take, whatever's outside and move it in. So we're forcing the situation where it's not at equilibrium. I'm putting everything in their places. All right. So it requires a pump, and it requires the sodium potassium pump to do that. An enzyme called ATPase, anything with ACE is an enzyme, okay, starts pumping things to their place. So it pumps 3 sodium outside of the cell to every 2 potassium in. And it's just moving them out. So let's throw the sodium out, let's put the potassium in, okay? Requires ATP, and it's really required for these impulses in nerve transmission. It's going to require it in the muscle cells too, but mainly because they're getting that information from the nerve cells. So we just stack it, basically. Just put as much of one type on one side of the cell and the other type of ion on the other side of the cell. We're not at equilibrium. We're going against the concentration gradient. Another type of active transport that doesn't include these little protein carriers is moving through the cell by using the cell membrane itself. So if you look at this picture here, notice this little cell membrane starts to, we call it invaginate. It starts to bend inwards, and it keeps bending inwards until it just pinches off and it forms this little vesicle on the inside. And really the outside of that vesicle is just part of that cell membrane that pinched in. This is called endocytosis. We're bringing something into the cell, and we're using the cell membrane to form a pouch called a vesicle by doing this. So the cell is actually physically surrounding, engulfing, and pulling something in. Okay, it's engulfing substances. That requires energy for the cell to actually seek out, surround, and pinch in a part of its cell membrane to become part of that vesicle. There's a couple types of endocytosis. First one is called phago. Phagocytosis is what I said earlier. The cell seeks out, surrounds, and pinches in and pulls in a particle, but it's specifically pulling in something that's solid. Okay, just like that little picture is showing you, pulling in something solid. Pinocytosis is the same thing, but it's liquid. So it's doing the exact same thing that you see in this picture, but instead of a solid little particle, it's water or something liquidy, okay? But it's pulling it in. The third type is a little more complicated. The third type of endocytosis, you need a specific protein receptor for this to occur. So this is this one right here. What happens is on that cell membrane, we have these little protein receptors, and you can see them, they look like the green hooks right here. They have to actually attach a particle to it. And when they get enough particles, little stars attached to it, it starts to cause an action where the inside bends in and eventually will pinch off into this. Unlike phagocytosis, where the cell actually just kind of surrounds it and pulls it in, this actually has to have an attachment of those particles first. So we have to have a binding before that substance can enter. That means not everything can enter. That cell membrane is not just going to surround any solid particle. It can only surround a specific particle that can attach to those receptors. So it's being a little more specific here. All of these still require energy from the cell to do this. Now, if I can bring something in, all of these bringing stuff in, right? The cell can also take things out. So exocytosis is endo in reverse. So it's taking something and it's expelling that out of the cell. So it takes a little vesicle that came from something like the Golgi apparatus, right? A product that the cell made or a waste product or something. And that membrane from the vesicle, what's blue, becomes part of the cell membrane. Remember, it's all phospholipids anyway, so it doesn't matter where it originated. It's still the same type of membrane structure, so it can pinch off or it can become part of the cell membrane again too. It doesn't matter. It's still the same substance, but we're going out of the cell instead of inside of the cell. This happens commonly at the ends of neurons when chemicals called neurotransmitters are released out of the cell. So a product that is required to initiate another reaction on a different cell, neurotransmitters are released by exocytosis. Now, I brought things in with endo. I took things out with exo. Now I can do both. I'm jumping ahead here. Here's a Golgi apparatus. So again, bringing that out. There's the vesicle. The vesicle becomes part of the cell membrane and it releases. That's exocytosis, right? Coming in, that's exo too. But if I brought something in, it would pinch off this way and then it would become a little vesicle here. That's endo. And then you have the two types, phago and pinocytosis. This is what I was trying to get to when I jumped ahead. Transcytosis is a combination. You have endocytosis, bringing something in, and then you immediately expel it on the other side of the cell. So here's endo, whatever it is coming in, white blood cells bringing something in here, and then that thing travels through the cell and it's released on the other side of the cell with exocytosis. So trans is just bring it in and then take it out again. Unfortunately, fortunately for us, this is something that we can do, our cells can do for like absorbing nutrients and things across the cell lining. So we need this to happen. Unfortunately, you can have pathogens like HIV that causes AIDS can sort of hijack this mechanism of the cells and can cross through barriers in layers of cells like the vaginal wall or something, pass through those cells right there and into your bloodstream through that same process. So it can be done, normally, but it also can be used for other things, can use it and hijack that process. And that's going to be it for this section. We'll talk about cell division in the next section.